A few years ago, a study crossed my desk and made me stop cold, not because of what it said exactly, but because of what it implied. Researchers had analyzed the gut microbiome of patients with colorectal cancer and compared it against healthy people of the same age. The difference wasn't small, it wasn't marginal. It was a gap so pronounced that scientists could predict with high accuracy who had cancer simply by looking at the bacteria in their stool. No colonoscopy, no biopsy, just the microbial profile. And what disturbed me most wasn't the finding itself. It was the question that finding left hanging in the air. How many of those differences come before the cancer, not after? I'm Dr. Becker, and I've spent years investigating the connection between what happens in the gut and what happens everywhere else in the body, especially in people over 50. What I'm about to share comes from that uncomfortable intersection of preventive medicine, emerging oncology, and microbiology that is rewriting what we thought we understood about how the body detects and destroys abnormal cells. I'm not going to promise you a cure. What I'm going to do is walk you through the evidence, explain the mechanisms, and give you a concrete protocol grounded in what the most recent research actually says. Today, you're going to understand why certain gut bacteria are capable of powering the immune system's anti-cancer response, how natural aging specifically depletes those bacteria at exactly the moment you need them most, and precisely what to eat to reverse that process. By the end of this video, you'll have information that most physicians still aren't communicating systematically to their patients in their 50s, 60s, and 70s. And that, in terms of preventive health, can make a real difference. Before we go deeper, if you're finding this kind of research valuable, a subscription helps me keep investigating topics that matter. No pressure at all. I just mention it because your support shapes what I dig into next. Let's start with a number I want you to keep in mind. 50%. According to the broadest datasets we have, including European and American cohort analyses with hundreds of thousands of participants, approximately 50 to 60% of colorectal cancer cases, which is the third most common cancer after the age of 50, are attributable to completely modifiable risk factors, not inevitable genetics, not biological destiny. Diet, sedentary behavior, intestinal dysbiosis, chronic low-grade inflammation sustained over decades. And that means there's an intervention window that exists, backed by peer-reviewed research, that most people aren't taking advantage of, because nobody's explained it to them in terms they can actually use. Now, what the science of the microbiome has discovered in the last 5 years specifically changes how we understand that window. Because it turns out this isn't just about fiber and vegetables in the generic sense you've probably heard 1,000 times. It's about deliberately cultivating specific bacterial populations that have very precise, very well-documented immune functions. And those populations, in particular, decline with age in ways that coincide exactly with the increased oncological risk in the 5th and 6th decades of life. Let me take you through the research the way I found it, because I think the trail matters as much as the conclusions. The first thread I pulled came from a paper published in the journal Gut in 2023. Researchers were studying Akkermansia mucinifila, a bacterium that lives in the mucus layer of the intestine and that until relatively recently received little attention in oncological contexts. What they found was this: the bacterium produces a specific enzyme, an acetyltransferase, that has the ability to reprogram the tumor microenvironment in colorectal cancer models. What that means in plain terms is that the bacterium doesn't act directly on the tumor. It changes the conditions surrounding it, making the environment less hospitable for tumor growth. Think of it as changing the climate of a region so that a certain species can no longer thrive. The tumor doesn't die from a direct attack. It dies because its ecosystem was altered from within. Now, that was primarily an animal model study, intriguing enough to keep reading, but insufficient to draw definitive conclusions. So I kept following the thread. And what I found was a systematic review published in MPJ Biofilms and Microbiomes, a Nature Publishing Group journal, in 2025, synthesizing more than a decade of research on microbiome and cancer in humans. This review identified a consistent pattern across multiple cancer types and multiple populations. Patients who had higher levels of Bifidobacterium, Akkermansia mucinifila, and certain lactobacillus families in their microbiome showed better anti-tumor immune responses, specifically through the activation of a type of immune cell called the CD8+ T cell. and in patients receiving immunotherapy for advanced melanoma, lung cancer, and renal cancer, the presence of these bacteria predicted with notable consistency who responded to treatment and who didn't. That stopped me, because what this implies is not minor. It implies that the microbiome is not just a digestive system, it is a regulator of the immune system. And if you can modulate the microbiome, you can, to a meaningful degree, modulate the immune system's capacity to do its primary job, detect abnormal cells, and eliminate them before they become something clinically dangerous. There's a third point of evidence I want to mention before we get into the mechanisms, because it comes from a completely different direction and arrives at the same conclusion. In 2021, the Stanford University team published in the journal Cell a study comparing two diets over 10 weeks, one high in fiber and one high in fermented foods. The fermented food diet, which included yogurt, kefir, sauerkraut, kimchi, and similar foods, not only increased microbial diversity, it significantly reduced 19 biomarkers of systemic inflammation, including cytokines we know are involved in tumor promotion. 19. In 10 weeks. That's not trivial. And what makes that data especially relevant for people in their 50s through 70s is that chronic low-grade systemic inflammation, what scientists call inflammaging, is now considered one of the primary drivers of accelerated aging and increased oncological risk in the second half of life. Let me pause here to connect these three threads before we go deeper, because I think the connection is what makes all of this cohere as a system rather than isolated data points. We have specific bacteria that modulate the tumor microenvironment. We have specific bacteria that activate anti-cancer immune cells. We have evidence that concrete dietary interventions increase those bacteria and reduce the inflammation that favors cancer. And all of this is happening in the gut, where 70% of the entire immune system of the human body lives. That's not a coincidence of design. The gut is the largest immune organ in the body, and the bacteria living in it are its permanent instructors. Now let me explain exactly how these mechanisms work, because if you understand the why, the practical protocol I'm going to give you at the end will make far more sense, and you'll be able to follow it with real intention rather than just following blind instructions. The first mechanism is what I call the epigenetic guardian, and it involves a molecule called butyrate that you've probably heard mentioned, but may not know exactly what it does. When bacteria like Faecalibacterium prausnitzii, Roseburia intestinalis, and Eubacterium rectal ferment the fiber you eat, they produce butyrate, a short-chain fatty acid. Colon cells use butyrate as their primary fuel. That's already important. But what's extraordinary from an oncological standpoint is what butyrate does in cells that have begun to become abnormal. Butyrate inhibits a family of enzymes called histone deacetylases, or HDACs. And those enzymes are what compact DNA in ways that silence specific genes. Among the genes they silence are precisely the tumor suppressor genes, the genes that tell an abnormal cell to stop, not to divide, to die in an orderly process called apoptosis. Think of it as a security system that someone has deactivated. In a cell that's beginning to transform toward malignancy, the tumor suppressor genes have been silenced, essentially disconnected. Butyrate arrives and inhibits the enzymes that are keeping those genes disconnected. It turns them back on. The cell receives the signal again that something is wrong and activates its self-destruction protocol. This is one of the reasons the colon, which is in constant contact with butyrate produced by bacteria, has protective mechanisms that depend directly on microbial health. The specific problem for people over 50 is this. Faecalibacterium prausnitzii, which is the most important butyrate-producing bacterium in the human intestine, declines approximately 40% in older adults compared to younger adults. This decline is documented in longitudinal studies and is consistent across populations. And it coincides temporally with the period when colorectal cancer risk begins to increase most sharply. I'm not saying the loss of this bacterium directly causes cancer. I'm saying the loss of this bacterium reduces one of the surveillance mechanisms the body uses to keep cellular transformation under control, and that matters. The second mechanism is the immune army training, and this is directly connected to the immunotherapy data I mentioned earlier. 70% of the immune cells in your body live in the tissue surrounding the intestine. That's not accidental. The gut is in permanent contact with the external world through what you eat, and the immune system needs to be there to continuously learn what's a threat and what isn't. The commensal bacteria, the ones living peacefully in your intestine, actively participate in that teaching process. Specifically, they stimulate the maturation of dendritic cells, which are the cells that act as messengers between innate and adaptive immunity. Mature dendritic cells carry information to lymph nodes and activate T cells, including those CD8 plus T cells, which are the ones that literally identify and destroy infected or cancerous cells. Akkermansia mucinifila and bifidobacterium are the two actors that most consistently appear in the literature as stimulators of this CD8 plus response. In clinical trials in advanced melanoma, non-small cell lung cancer, and renal cancer, patients who had higher abundance of these bacteria before receiving anti-PD-1 immunotherapy showed significantly higher response rates. What that tells us, and this is crucial, is that the microbiome is involved not only in prevention, it's involved in the immune system's capacity to respond when a threat is already established. Your gut is training your immune system every single day. The question is whether it's training it well or poorly. The third mechanism is the barrier, and I want to explain this with an image I think makes the concept easy to retain. Think of the intestine as a series of very fine pipes. Those pipes are lined by a single layer of cells connected to each other by structures called tight junctions. When those junctions are intact, only what should pass into the body actually passes. Nutrients, water, electrolytes. When they deteriorate, things pass through that shouldn't. Specifically, fragments of dead bacteria called lipopolysaccharides, LPS, enter the circulation. And LPS is a potent activator of systemic inflammation. When the immune system detects LPS in the blood, it responds as if there's an active infection. It activates inflammatory pathways, releases pro-inflammatory cytokines, and puts the body in a state of constant low-grade alarm. That is the inflammaging I mentioned earlier, and chronic systemic inflammation, sustained over years or decades, is an established promoter of multiple cancer types, not just colorectal. It fuels the microenvironment that allows abnormal cells to survive and divide rather than being eliminated. Akkermansia mucinifila specifically has a critical role in maintaining that barrier. It lives in the mucus layer and actively regenerates it. When Akkermansia declines, the mucus layer thins, tight junctions weaken, and intestinal permeability increases. In older adults, this is especially relevant because intestinal permeability tends to increase naturally with age, and depleting Akkermansia through diet and lifestyle accelerates that process. The fourth mechanism I call the tumor epigenome. It's the most technical of the ones I'm going to explain, but also the most exciting from the standpoint of what it implies for the future of oncological treatment. There's a problem that T cells face when trying to attack a tumor. Tumors are very good at shutting down their attackers. They do it through a pathway called PD-1/PDL1. Basically, the tumor places a molecular signal on its surface that tells T cells, "Stop, you're not needed here, stand down." The T cells obey and become what oncologists call exhausted, functionally inactive. It's one of the primary mechanisms by which cancer escapes immune surveillance. Immunotherapy drugs like pembrolizumab and nivolumab work by blocking that signal, reactivating T cells. That's why they're called checkpoint inhibitors. And it turns out that butyrate produced by gut bacteria has a similar effect through a different mechanism. By inhibiting HDACs in the T cells themselves, butyrate modifies their gene expression in ways that reduce exhaustion and keep T cells functionally active for longer. A study published in 2024 demonstrated that in colorectal cancer models, inhibition of HDAC3 and HDAC8 by butyrate reactivated CD8 plus T cells that had been suppressed by the tumor. The microbiome was literally unchaining the immune cells the tumor had put in chains. This is the mechanism that I believe will most transform oncology in the next 10 years. Not as a substitute for checkpoint inhibitors, but as a complement, a way to potentiate the response to those treatments through something as accessible as what we eat. The fifth mechanism, and with this I finish the biology section before moving into the practical protocol, is what I call the pathogen ecology. The intestine is not only home to the good bacteria, it also houses bacteria that, when they grow disproportionately, produce substances that promote cellular damage. Fusobacterium nucleatum is the most studied of these. It's found in high concentration in colorectal tumor tissue, and studies show its presence in the tumor is not just a marker, it's an active participant. It produces proteins that inhibit apoptosis of cancer cells and promote immune evasion. Toxigenic Bacteroides fragilis, certain colibactin-producing strains of E. coli and other potentially harmful bacteria are also documented as pro-carcinogenic in the intestine. What's relevant here isn't memorizing them, but understanding that microbiome composition is an ecosystem. When beneficial bacteria like Faecalibacterium, Akkermansia, and Bifidobacterium are abundant, they act as a competitive barrier that prevents the overgrowth of the harmful ones. When the microbiome deteriorates, that barrier disappears, and the opportunists take over the space. So now you have the full picture. The good bacteria produce butyrate that activates tumor suppression mechanisms and keeps T cells active. They train the immune system to be more effective. They maintain the intestinal barrier intact and reduce systemic inflammation. And they occupy the competitive space so harmful, bacteria can't gain ground. All of this happens in your gut, every day, depending in large part on what you choose to eat. What should concern you, if you're over 50, is this. All of those systems tend to deteriorate with age in ways you don't feel. They don't hurt. They don't produce immediate symptoms. They deteriorate silently over years while oncological risk increases in the background. But there's another side to that story. And it's that the studies also consistently show the microbiome is extraordinarily plastic. It responds to dietary changes in weeks, not years. And that means the intervention window is open, and it can be opened relatively quickly. Here is the protocol, based on the evidence we've reviewed and on what clinically makes sense for the 50 to 70 age range. The first priority, and the most impactful, is reaching 30 grams of fiber per day from diverse sources. The American Institute for Cancer Research specifically recommends this amount for colorectal cancer prevention. A meta-analysis examining data from the European Prospective Investigation into Cancer and Nutrition, a study with hundreds of thousands of participants followed for over a decade, found that every additional 90 grams of whole grains per day was associated with a 17% reduction in colorectal cancer risk. 17%. And the mechanism is direct. Fiber is the food of butyrate-producing bacteria. What matters isn't just the quantity, but the diversity of sources. Different types of fiber feed different bacteria. Soluble fiber from oats and legumes feeds primarily Bifidobacterium. Inulin from garlic, onions, asparagus, and artichokes is especially effective for Akkermansia and Lactobacillus. Resistant starch, found in green bananas, rice and potatoes cooked and then cooled, and raw oats, feeds selectively the butyrate-producing bacteria like Faecalibacterium and Roseburia. How to implement this practically? Oatmeal at breakfast with ground flaxseed is already 12 to 15 grams. A bowl of lentils or chickpeas at lunch adds 10 to 12 more. An apple with the skin, some asparagus or artichoke at dinner, and you're at 30 without extraordinary effort. The most common mistake I see is trying to go from 5 grams to 30 in a week. That produces gas, bloating, and abandonment. The right approach is to increase by 5 grams per week over 4 or 5 weeks. The intestine needs time to adapt, and if you do it gradually, the symptoms are minimal. The second priority is plant diversity. There's a figure from the American Gut Project, the largest human microbiome study to date, that is counterintuitive but very robust. People who eat more than 30 different types of plants per week have significantly more diverse microbiomes than those who eat fewer than 10, regardless of the total quantity of vegetables consumed. Diversity matters more than quantity of any single thing. What counts as a plant? Any vegetable, fruit, legume, grain, nut, seed, aromatic herb, or spice. Garlic is a plant. Turmeric is a plant. Black pepper is a plant. A bowl of curry with brown rice, chickpeas, garlic, onion, tomato, turmeric, cumin, and cilantro is 8 plants in one meal. If you keep track for a week, most people are surprised by how close they already are to 30 without doing anything extraordinary. And those who fall far short tend to be the ones eating the same 3 or 4 vegetables repeatedly. The third priority is introducing fermented foods regularly. This is where the Stanford evidence I mentioned becomes directly actionable. The study found that 1 to 6 servings per day of fermented foods, not supplements, but real fermented foods like plain unsweetened yogurt, kefir, sauerkraut, kimchi, miso, and tempeh, produced consistent effects on microbial diversity and inflammatory reduction in 10 weeks. Fermented foods don't just deliver live bacteria to the gut, they also deliver metabolites already produced by those bacteria during the fermentation process, including lactic acid, bioactive peptides, and compounds with direct anti-inflammatory effects. Kefir specifically contains a far more diverse range of strains than most commercial yogurts. Unpasteurized sauerkraut and kimchi have active bacterial colonies that pasteurized versions don't. When you buy these products, check that the label says contains live cultures or unpasteurized. 1 to 2 servings a day is sufficient to see effect. Half a cup of yogurt at breakfast, 2 or 3 tablespoons of sauerkraut with lunch. You don't need large quantities. You need consistency. The fourth priority is reducing or eliminating the 2 food groups that most consistently associate with dysbiosis and colorectal oncological risk. The first is processed meats, deli meats, bacon, sausages, hot dogs, cured meats. The International Agency for Research on Cancer, the WHO's Cancer Evaluation Arm, classifies them as Group 1 carcinogens for colorectal cancer. That's the same level of evidence as tobacco for lung cancer. The mechanism is multiple. Nitrates and nitrites used as preservatives convert to N-nitroso compounds in the gut that are directly mutagenic. Heterocyclic amines formed during high-temperature cooking damage cellular DNA, and processed meats selectively suppress butyrate-producing bacteria while favoring the growth of fusobacterium. I'm not saying an occasional ham sandwich causes cancer. I'm saying regular, frequent consumption of these foods over decades has a cumulative effect on the microbiome and oncological risk that the evidence doesn't allow us to ignore. Unprocessed red meat is a different and more nuanced case. The AICR recommendation is less than 500 grams of cooked red meat per week, with preference for preparations that don't involve direct flame cooking or very high temperatures that generate heterocyclic amines. The second problematic category is ultra-processed foods in general, those containing industrial emulsifiers, carrageenan, polysorbates, which recent studies in animal models and human observational data show specifically alter the intestinal mucus layer and reduce Akkermansia abundance in targeted ways. It's not coincidental that the modern Western diet, high in ultra-processed foods and low in fiber, correlates so consistently with impoverished microbiomes and elevated oncological risk. The fifth priority is paying attention to 2 factors that affect the microbiome in ways most people don't consider: antibiotics and proton pump inhibitors. Antibiotics are sometimes necessary and life-saving, but a recent analysis showed that a single course of antibiotics can reduce Faecalibacterium prausnitzii by up to 60%, and the microbiome may take 8 weeks or more to recover, and in some cases doesn't recover completely. If you need to take antibiotics, reinforce your intake of fermented foods and prebiotic fiber before, during, and after the treatment. It doesn't cancel everything, but it minimizes the damage. Proton pump inhibitors, drugs like omeprazole used for reflux and ulcers that millions of people over 50 take chronically, have an effect on the microbiome comparable to antibiotics, according to a 2023 meta-analysis. They reduce gastric acidity, which is a natural defense mechanism against colonization of pathogenic bacteria in the small intestine. The result in chronic users is a less diverse microbiome with higher prevalence of undesirable bacteria. If you're taking these drugs regularly, the conversation about whether you still genuinely need them is worth having with your doctor. The sixth area of intervention is not nutritional, but the evidence on it is solid enough to include: exercise increases microbial diversity in ways that are independent of diet. An analysis of multiple studies found that physically active adults have greater abundance of butyrate-producing bacteria than sedentary adults with similar diets. The probable mechanism involves changes in intestinal transit time, colonic pH, and circulating cytokine levels that modulate microbial ecology. The type of exercise that most consistently appears in the literature as beneficial for the microbiome is a combination of moderate aerobic exercise, brisk walking, swimming, cycling, with light resistance training twice a week. You don't need extreme intensities. In fact, very high intensity exercise can have transient negative effects on intestinal permeability. What works is regularity at moderate intensity. Sleep deserves a brief mention as well. The microbiome follows circadian rhythms, and chronic sleep deprivation, fewer than 6 hours regularly, is associated in observational studies with dysbiosis and increased inflammatory markers. The mechanisms involve the gut-brain axis and cortisol dysregulation. For people over 60, who frequently experience deterioration in sleep quality, this adds another layer to why sleep is not a luxury, but a health intervention. Now I need to say something about supplements because it's one of the questions I receive most often, and I think the honest answer is not the one the industry wants you to hear. Probiotic capsules have limited and heterogeneous evidence for cancer prevention. The studies that show the most consistent benefits from probiotics use them in specific contexts. Reduction of post-surgical complications in colorectal cancer patients already being operated on, improving side effects of chemotherapy, post-antibiotic recovery. The evidence that taking a lactobacillus capsule as a healthy adult prevents cancer is honestly much weaker than the evidence that eating a diverse diet rich in fiber and fermented foods does so. Prebiotics, on the other hand, like inulin or PHOS available as supplements, have more solid evidence in studies showing increases in bifidobacterium and improvements in intestinal barrier function. If the diet can't cover the needs, a supplement of 6 to 10 grams of inulin per day is a reasonable option with evidence. But I'd rather my patients get it from artichokes, onions, and garlic because those foods come with dozens of other bioactive compounds that isolated inulin doesn't have. Resistant starch as a supplement has some interesting emerging evidence. High maize cornstarch has shown in some trials a specific ability to increase butyrate-producing bacteria. But again, green bananas, cooled cooked rice, raw oats, these are accessible sources with the same effect and additional benefits. When should you expect to see results? Microbiome studies show that changes in composition are detectable within 4 to 6 weeks of consistent dietary change. The first 2 weeks, what you'll notice most are changes in bowel habit, possibly more regularity, possibly some gas at the start if the fiber increase was too fast. In the first month, inflammatory markers begin to improve in people who start from elevated levels. In 2 to 3 months, microbial diversity increases in a more established way and fecal butyrate levels are higher. The benefits on oncological risk are by definition long-term and not directly measurable in an individual. But they're the ones epidemiology demonstrates in populations with consistently richer fiber and fermented food diets over years. I have to be honest about something the science in this field is still resolving. The causality between microbiome and cancer in humans isn't fully established in every aspect. We know there's a strong and consistent correlation between certain microbial profiles and oncological risk. We know the biological mechanisms by which good bacteria protect are plausible and documented in animal models and in vitro studies. We know dietary interventions change the microbiome in ways that go in the right direction. But the prospective long-term RCTs that directly demonstrate that modifying the microbiome in a 50-year-old reduces their cancer risk 20 years later don't exist yet, because those studies take decades to complete. What does exist is enough convergent evidence that the interventions I've described are reasonable, low risk, with multiple other benefits beyond the oncological, and backed by the leading cancer research and nutrition organizations in the world. And there's one more thing I want to say, directed specifically at people in the 50 to 70 range, the audience of this video. There has been a medical narrative that for decades emphasized early detection as the primary strategy against cancer. The colonoscopy, the PSA, the mammogram, all of that is valuable and important. Keep getting the screenings recommended for your age. But early detection means the process has already started. Primary prevention, modifying the biological environment of your body so that process has less chance of starting, is a conversation that preventive medicine has been slow to have with sufficient specificity. What microbiome research is doing is giving us that specificity, not at the level of eat more vegetables, but at the level of which specific bacteria, which exact immune functions, which metabolites, which dietary interventions produce which measurable changes. That's what I find genuinely exciting about this field. And it's why I dedicated this video to explaining it with the depth I believe it deserves. If you have symptoms we haven't discussed today because they're beyond the scope of this video, blood in the stool, persistent change in bowel habit for more than 2 or 3 weeks, unexplained weight loss, persistent abdominal pain, intense unexplained fatigue, those symptoms require immediate medical evaluation, not the protocol I just described. Medical evaluation. The microbiome is preventive, not therapeutic when symptoms require diagnosis. Likewise, if you have an active diagnosis of inflammatory bowel disease, Crohn's, or ulcerative colitis, if you're in active oncological treatment, if you're immunocompromised for any reason, talk with your doctor before implementing significant dietary changes or adding supplements. In some specific clinical contexts, what's beneficial for most people may require modifications. And if you take anticoagulants like warfarin, be aware that gut bacteria produce vitamin K, which interacts with that medication. Significant changes in the microbiome may require adjusted monitoring of your INR. We started with a study that could predict cancer through a microbial profile, and we've arrived here. An ecosystem of bacteria in your gut that is training your immune system, producing metabolites that activate tumor suppressor genes, maintaining the barrier that prevents systemic inflammation from becoming chronic, and competing for space with the bacteria that promote cellular damage. That ecosystem is plastic. It responds to what you eat. It responds to exercise. It responds to sleep. And it declines with age precisely in the decades when you need it most, which makes the interventions we've discussed today not optional, but something with a serious, well-documented preventive argument behind them. This is not genetics you can't change. It's ecology. And ecology responds to the conditions you create. The fact that you're here seeking this information, going deeper into the mechanisms rather than just looking for magic recipes, already puts you in a different position. You have more control over the biology of the second half of your life than you've probably been told. And what I've shared today isn't speculative theory. It's peer-reviewed evidence published in Gut, in Cell, in Nature, at the American Institute for Cancer Research, being discussed in oncology, gastroenterology, and preventive medicine, and informing the guidelines of the world's most rigorous health organizations. Leave in the comments your biggest question or takeaway from today. I read all of them, and they guide what I investigate next. Keep investigating, and I'll see you in the next one. Dr. Becker.